Good morning, everybody. It's a pleasure to introduce this colloquium, this mini colloquium on non-equilibrium physics, in biology. So in particular, the idea of organizing this mini colloquium, I must tell you a bit of details. This was done after some conversations with Kees van der Beek from Paris and Christian Beck from London, with whom we share some, let's say we have some, we're sharing some of the sections in the European Physical Society, and in particular, I'm chairing the session, or I'm chairing the section on, or the division on physics in biology. And then we were realizing the many connections that there are between biophysics and condensed matter physics. So the idea was to try to organize in this big meeting is organized now in Madrid. The idea was to set a mini colloquium where we try to connect or to put in relationship the many applications of physics of condensed matter when applied to biology. And what we have been doing, it has been to prepare a mini colloquium dealing basically on systems out of equilibrium, ranging from active systems to, for example, fluctuations in small systems, stochastic thermodynamics, networks in biology, cellular systems, and so on. So I think that this can be very interesting mini colloquium. Unfortunately, cannot be done in real life. It has to be done online, which is not something I really like, but that's the current situation. So with this, I want to finish this introduction, this brief introduction. I hope you will enjoy the many talks we have put together. I must thank especially Laura Rodriguez and Juan Aragonés, and of course also Timon Idema, who have contributed a lot in making possible this panel of excellent and outstanding speakers. So thank you very much, Laura, thank you very much, Juan, and thank you very much, Timon. And with this, I give my word I pass the word to Iker Thurigel. So Iker Thurigel, just a few words about him. He made his PhD, his doctorate, and well, actually his PhD, okay, and in granular systems with Tom Molin in Pamplona in the University of Navarra, and then lately he has been turning his interest from, let's say, studying granular systems, which are, if you want, non-living systems somehow, but they have many connections to active matter, which, as you know, it's a subject of intense research at this moment. So he switched actually from granular systems to active matter, but in particular to what we now understand as macroscopic active matter, which is big particles that are propelled by external sources or internal endogenous sources of energy. So he did several postdocs in Bordeaux and Manchester, and right now he's at the same place where he started, at the University of Navarra, where he was, let's say, approved for a full professor position. And he will talk today about the flux of discrete systems, granular colloids, for example, or other bigger objects that are relevant for biology across narrow channels, for example, like ion channels in membranes, okay, which is of much relevance, as you know, in biophysics. So thank you very much, Iker, for accepting our invitation to deliver this talk, and I pass you the word. Thank you very much.
Okay, thank you, Felix, for these nice words. As you say, yes, perhaps I'm sharing the screen now. As you say, perhaps this is a very strange talk for this meeting, because I guess that the biological systems you are expecting are on the micro scale or nanoscale. Today I'm going to talk about biological systems mostly, but at a different scale. Even though I think that perhaps some of my data, some of my results can be inspiring for you to apply to other different systems. Let's see what you think. Okay, so what I'm going to talk is what happens with discrete systems. So these systems of many particles when they flow through constrictions through narrow channels. So let me start with an example. It's a funny example. It's very, very illustrative of what I'm going to explain today. This is a group of sheep. Here you have the sheeper, and then when the sheeper is going to open the door because the sheep want to get feeded, so the sheep start to flow through the narrow door, and what happens? This green line is a line that we take here to build a spatio-temporal diagram, and when there is this flow of sheep, here you can see very nicely in the spatio-temporal diagram what is going on. What is going on is that the sheep flow, but suddenly there is a block at the door, a geometrical block. Then we have this kind of intermittency in the flow. You have flow, then you have a clog here because there is this empty space means that there is no flow. And again, here the same happens, etc. So this is, the question is if this is interesting for something, if it's interesting for in other systems. And the answer is, I think, is yes. And this kind of behavior of blocking or clogging, flowing may happen in many different systems. The simplest perhaps is just a granular system where you have every single day you shake the salt cellar to pour salt in your food. So this is what happens. You have to shake the salt cellar to avoid this kind of clogging. The same may happen in blood vessels, even in pedestrian evacuations with panic, evacuating, for example, a stadium. Or even in microbial populations, when they flow through a constriction, you can have this kind of intermittent flow, which of course depends on the kind of constriction. Okay, so among all these systems, perhaps the most strange, I will say, from a physical point of view, or the most different one, is the granular system. And the reason is that this system is not alive, is not active. The other ones somehow may have some kind of input of energy, like for example, blood vessels, the flow of plasma, but in these cases, in all these cases, you may have intermittent flow. In this case, the flow is not intermittent at all, because when you have a clog in a silo, in a granular system, the clog will resist forever. I mean, there is no internal energy. Proof is a very nice proof in the world that is this immortal bridge in China, which is living there for centuries. You need an earthquake or you need something to shake this clog, to break the clog and have intermittent flow. This is what we do in the lab. In the lab, what we do is to shake the whole system. And even if the shaking amplitude is tiny, these particles are millimeter size, even if the shaking is, I would say, less than a micron amplitude, what we have is this. Okay, we have a clog, and then you cannot see the shaking because it's tiny, but the flow is intermittent again. Okay, there will be flow, and eventually there will be a clog, and flow, and clog, and so on and so forth. So, okay, until of course the silo is emptied. So, okay, the system is characterized by these flowing times, clogging times, flowing times, clogging times that alternate. So now we are going to analyze the statistics of this behavior, the statistics of these flowing times and clogging times. And what we observe is a very interesting scenario. The flowing times, note that the flowing times are telling us about probability of getting a clog form, okay? Because you have a flow interval, and eventually there will be a clog that will arrest this flowing time, right? So what we have here in the flow intervals is an exponential distribution. This happened in the distribution of this logarithmic scale. So the flow intervals are exponentially distributed. This is very interesting because, well, interesting, no, it's boring indeed, because what we have is a characteristic time. We have something that is a Poisson process at the end of the day. It's a constant probability of getting a clog. Okay, exponential distribution means that the probability of getting this clog form is always constant during the whole process of the avalanche or during the whole process of this flow. So this is boring. scenario, and we have seen this in many different systems. However, the clocking interval is telling us about the clock destruction. Okay, when you have this clock, the system managed to destroy this clock for different reasons in different systems. So what we find here is that the clocking intervals are distributed with a distribution that is broad tails. Okay, the tails are broader than in the case of the flowing intervals. Indeed, in this original paper of Handa et al. In a silo as well, different one, but it doesn't matter here. They suggest that this was power loss. Okay, so this is the first bit of information that was important to tell today, is that the clock formation is a process that is memorylessness, is a kind of Poisson process, constant probability of getting a clock, okay, because you have exponential distribution. However, the clock destruction process, okay, is characterized by distributions with broad tails in the clogging intervals. So this is a history-dependent process. Something is happening in the arcs, in grains, in other systems we will see now. Something is happening that the process of how the clog, this structure that is arresting the flow, evolves is not memorylessness. There is some kind of history dependence there. Okay, so now this was observed with grains more than 10 years ago. The question now is this generic? Is this, can you extrapolate this to other systems? Because at the end of the day is what is interesting here, right? So the last work I've been involved with is in a suspension of particles with fluid. Here there is not shaking at all, it's just fluid particles. There is a clog formed, and then the fluid passes through the holes of the particles, and this fluid passing through the particles 100 microns, more or less, is the one that is destroying the clogs. Okay, so I'm going to put again kind of the same kind of movie, and if you see here, this is the line I'm taking or we are taking to build this spatial temporal diagram. And again, we have this kind of the same behavior. Okay, we have this flowing, clogging, flowing, clogging, flowing, clogging, alternation of flow and clog intervals. So again, we can measure the flowing intervals, and we have an exponential distribution of the flowing intervals. It doesn't matter if you take, if you count them by number of particles, or if you count the duration of the burst, the duration of the flows with the time that the system is flowing. In both cases, you have this exponential distribution that tells us once more about this Poisson process, constant probability of getting a clog form. And however, the arrest intervals, the distribution of these intervals of arrest are broad tail distributions. Again, we find something similar to a power law. Okay, the same happens with colloidal micrometer size simulations. Okay, this is a simulation of colloids with Ignacio Pago Navarro's group again as well. So you can see the intermittencies in the passage of these discrete systems. So sometimes, for example, now you have seen a clog and the clog is resumed. The same happens with centimeter size self-propelled particles or X-books. This is something that your kids perhaps may use to play with. It's very funny. So if you look at the intermittencies when you make them pass through a bottleneck, you observe that here there is this kind of clog. I'm going to put it again. Clogging, flowing alternation. Okay, I'm not going to show the distributions here, but again, you can go to the references and you will see that you have this exponential distribution of flowing intervals, power law distribution of clogging intervals. And finally, you can do the same with pedestrians, and again, you have the same. Okay, so this is the kind of problem I'm looking at, comparing different systems, many different systems, as you can see. And here you have the people passing through the door, and here you have had a clock. But of course, here in this system, we cannot reach clocks of few seconds. 2-3 seconds are maximum. Of course, we can be injured otherwise. People can be injured otherwise. But again, you can see here the intermittent behavior. Here you have a burst, and you have some tiny clocks here, and a burst, so on and so forth. And if you look at the behavior here again, exponential distributions for the burst sizes, power load, broad tails at least for the clogging intervals. So, okay, let me go back to the granular example, which is a kind of the easiest to have a lot of statistics, and tell you one thing, extra thing that is more important from my point of view, is that in these power load tails, in some situations, you may find that the exponent of the power load is above 2 for these two cases, and below 2 for this case. Depending on the parameters, depending on this scenario you are analyzing, you may find a power load for the clogging intervals. Remember, I'm just studying the clogging intervals now, which is above 2 shorter times, or below 2 in this case, which are longer clogging times. Okay, so this is important because what happens is that if the power law has an exponent which is below 2, you cannot compute the average. You cannot do the integration of this distribution to compute the average, because it does not converge. Okay, so if it does not converge, what it means is that if you, in the laboratory, if you measure during 1 hour, you will find average time that is smaller than if you measure for 3 hours. The average time will be, of the clogging time, will be larger and larger and larger as you measure longer and longer and longer. So it's a non-convergent distribution. Okay, so this is what we have used. We have used this feature of the distributions to define this flowing parameter, what we call flowing parameter, that is the order parameter in our scenario. In our transition from clogging to no clogging is going to be our order parameter. And it's very simple. I mean, flowing parameter is just the proportion of time that the system is in a flowing condition. So this is the average flowing time divided by the total time, which is the average of the flowing plus the average of the clogging time. And then what happens with this parameter is that this number will go to is well defined if the distribution, if alpha, the exponent of this distribution of the clogging times, is higher than 2, because in that case you can compute the average clogging time. Otherwise, if the exponent is 2 or lower than 2, you cannot compute this. This number grows unboundedly as you increase the window of time measurement. So in that case, if the exponent alpha is below 2, this flowing parameter will tend to 0, because this number grows unboundedly. Okay, so this is important. Huh? Okay, I'm going to sketch it. It's a bit tricky, so I'm going to do a sketch. Imagine that are 4 flowing clogging scenarios. The first one is very simple. It's all the time the system is flowing. So this order parameters will be 1. This is a flowing regime. When we have intermittencies, if the exponent is higher than 2, then you can compute this clogging time, the average clogging time. So this number is well defined and will be in between 0 and 1, because it's more than 1 because there are some intermittencies, but it's larger than 0 because these numbers are integral numbers. Okay, so what happens is the change is when you have a very similar behavior of intermittency, but the exponent alpha is below 2. It's not 2, it's below 2. This is the important change. In this case, the order parameter is 0. The flowing parameter tends to 0 when you start increasing the window measurement. So this is what we are going to call clogged regime. Although it seems intermittent, it is clogged because this order parameter is 0. And of course, in this case, when you have just few particles flowing and a clog that lasts forever, this will be clogged. So now the transition in this system, the transition will be from here, from this region of clogged to this region of unclogged. Okay? So this is what we are going to analyze, either with the order parameter or either with just looking at the exponent alpha. Alpha below 2 is when my system is clogged. Alpha above 2 is when my system is not clogged. It's either intermittent or flowing. We are not going to study this transition here. from intermittent to flowing, but we are going to study this transition from clogged to intermittent. Okay. So we did that with silos. I'm following the same scheme that before. I'm going to start with silos and then move to other systems. And with silos, well, you can do as many statistics as you like. So we are going to study three variables here: the height of the layer of grains above the outlet. So we have a silo, we have a system with grains, and then we can depending on the number of particles in the silo, we can control the height of the layer of grains above the outlet. We can also control the acceleration of the external vibration we are inputting in the system, and we can control the outlet size. So how is the transition? How we can move from this clogged to unclogged? Of course, we can move from clogged to unclogged, from clogged to unclogged by just increasing the outlet size. Okay, here alpha equal 1.7. This system is clogged. This corresponds to a small outlet, and then as we increase the outlet, we move to an unclogged scenario because we cross from alpha below 2 to alpha above 2. It makes sense. The larger the orifice, the more difficult to get the system clogged. Good. Next, excitation. Of course, as you increase the excitation, here the excitation is low, here the excitation is high. As you increase the excitation, you move from a clogged scenario to an unclogged scenario. Increasing the excitation helps the system to flow. And finally, this is a tricky parameter, which is the height of the grains that in the silo. Surprisingly, what happens is that if you have just a thin layer of grains in the silo, in that case, is easier, in this case, is easier to break the clogs. However, when you have a lot of grains on top of the clog, on top of the arch, it's more difficult to break the arch. So the transition is quite important here, just by tuning the height of the layer of grains. Here you have a lot of grains on top of the silo, so the clogs last longer than when you have a thin layer of grains, which is kind of counterintuitive, right? So this was encompassed in this idea of these three parameters in a more generic behavior that was just a proposal inspired by a paper of Mike Cates in PRL. So we say that we have in our system to do this transition from clogged to unclogged, three parameters that are very generic. The first one is a characteristic length. Of course, if you increase the outlet size, you increase this characteristic length in the system, and then you can unclog your system. Also, you can put some kind of energy in the system, which is called incompatible load. That is a name that has been taken from Mike Cates' paper. Then the incompatible load means that it's a load that you apply which is not compatible with the forces that your system has developed. And finally, you can unclog the system by reducing the compatible load. What is the compatible load? The idea of Mike Cates was 20 years ago was that the response of your system to the load that is driving your system. In my case, with the grains, we have the gravity. We have the gravity that is driving the system. So the arch that is formed at the outlet is responding to this gravity and is forming an arch. So if you are able to reduce the weight in your system, you are able to unclog the system, to remove this clog. So this is a bit more complicated. I'm going to put more emphasis on this compatible load later on. So is this generic? Again, this is an idea. We have compatible load, uncompatible load, and a characteristic length. Is this generic? Okay, I'm going to put just a summary of all the many systems we have been working with, of course, with collaborations with many different people. And here, first of all, we have granular silos, we have colloids, we have colloid simulations, we have sheep, we have pedestrian, we have robots. Outlet size, of course. Outlet size, door size. In all the cases, if you increase the outlet size, you can move from a system that is clogged to a system that is unclogged. You can favor the flow increasing the outlet size. We have proved that in many different systems. Well, note here different colors. When this white is because we have proved the transition. We have proved alpha above and below 2 in these cases. When there is blue, we have just seen a change in the exponent. We have not been able to reach the clogged situation, and the reason is because it's cheap and pedestrian. We cannot force these systems to this kind of scenario where to avoid injuries, of course. So, okay, the characteristic length is clear, is something expected. Also, the external excitation, that can be very different systems. Like, for example, granular, we have an external global vibration that we are inputting. We can have also a local vibration in a granular just with a piezoelectric, a very local shaking at the outlet precisely. Also with colloids, we can increase the temperature, the room temperature, and we can cross this transition. This was proved by simulations with Raúl Cruz and Ignacio Pavona Barraga. So it's proved that this incompatible load, this noise, this external excitation, internal excitation is helping to favoring the flow. And finally, we have this compatible load. This compatible load have focus in this system, and in a silo, we checked, we proved, as I said before, that reducing the layer of the grains favors the transition, but also we can incline the silo instead of being vertically aligned, we can tilt it a bit. And when you reduce, instead of being vertical, it's a bit tilted, reduce the gravity, you favor the transition, you favor the flow by reducing the gravity. And also with sheep and pedestrian, I will talk about this later. If we reduce the competitiveness of these guys, of these bodies, of these humans or animals, you favor the flow, reducing the competitiveness, reducing this load that is this driving force. And with robots, we also prove that reducing the crowd size helps to avoid clogging. So I'm going to focus on this for the last 5 minutes of my talk. I'm going to talk about pedestrians, for example. In pedestrians, what happens? What happens is something that was already known in pedestrian community, and it was called faster is slower. And what happens is that here I'm going to put the same video that before, that was for high competitiveness, but now I'm putting nearby this low competitiveness video. Okay, so you will see the difference. Here. Okay, good. 5 minutes. Yes, 5 minutes will be enough. So what you can see here is very. Sorry, let me stop it. Now, okay. What you can see here is that when the people is just not pushing, the flow is very smooth. The spatiotemporal diagram is rather regular. There is not big clogs. There is not gaps here in the flow of people. So the clogs are almost non-existent. Okay? However, here, when people is pushing, apart from the behavior, nice behavior that you can find here, what you find here in the spatiotemporal, as I told you before, is this kind of clogs. And then if you go and look at the distributions of these clogging times, this is, instead of doing the normal histogram, I'm doing the survival function, but it's the same. Here you can see this kind of, well, power law with an exponent of 7 is kind of, but it doesn't matter. I mean, the important thing here is that the tail gets broader as you increase the competitiveness, okay? The clogs last longer as you increase the competitiveness, okay? This was known for pedestrian. It was not proved, but was known by simulations in pedestrian community, and the term was called faster is slower. So we say, okay, good, we have proved that. Increasing the competitiveness, we have more clogs, good. Now, because we increase the compatible load, and this is not good for the flow. So with the XBAPs, with these robots that self-propel, we prove the same, but instead of with the competitiveness, because they move always at the same velocity, by changing the crowd size. With using by 21 agents, we find a power load with an exponent of 3.2. And when we increase the crowd size to 61 agents, we saw that the the tail got broader again, and the exponent was 1.9, we crossed the transition from not clogging to clogging. Okay, so in my opinion, this phenomenon that is known that faster is slower should be renamed, and it's not faster. The problem is not going faster or not faster. The problem is to be stronger or not stronger. So stronger is slower. Okay, if you push, if you increase the compatible load, the system will go slower because the number of clocks increases. You can see a comparison here. Here you find more clocks of 100 seconds, and in this case, as you increase the number of ages. So, good, we have an explanation for this well-known phenomenon in pedestrian dynamics, which is compatible with our scheme of a phase diagram. And finally, let me just a couple of minutes talk you about the role or the curious effect of placing an obstacle on top of the outlet. So what do you expect? In principle, if I ask someone, my mom, for example, what happens if you put an obstacle in front of a door? The flow is better or is worse? Well, the flow is worse, no? This is what my mom will say. Perhaps after this talk, you say, no, perhaps if we place an obstacle in front of a door, we reduce the compatible load because the obstacle is absorbing this load of the grains on top. We reduce the compatible load and the flow is better. Okay, you are right. This is what happens. If we place an obstacle on top of a silo, the flow here is better because we create here an empty region. The pressure here is not as high as here, and then the arches are not stabilized here. Okay, so it's more difficult to find an arch stabilized. Indeed, for a static silo, it's amazing. I mean, you may find this is the average number of grains that go out of the silo. This is the silo without the obstacle. With the obstacle, you may increase, for the optimal position of the obstacle, the avalanche size, the number of grains, by more than one order of magnitude. Instead of having the grains flowing for 10 seconds, you go to 1,000 seconds. The first time I did that in the laboratory, I couldn't believe that. I couldn't believe that. So, okay, the effect of the obstacle in grains is proof, it's very good. We also proved that with ship, and I will show it very quickly. So again, same videos than before. I'm going to back to the first video, but now comparing width and width of obstacle. What happens is that the flow is very similar. The average flow is very similar. There is a bit of improvement with the obstacle. But what is more important here is that with the obstacle, we reduce the clogs. This big clog, this big clog, this big clog. Here, when there is a clog, there are some clogs, the clogs are resumed very quickly. So the obstacle is helping in facilitating the resuming the flow. So, good. What happens with pedestrian? Because after grains,
you have to finish here.
Yeah, I'm finishing. After ship, we proved with pedestrian. This is the flow with pedestrian. We did 3 experiments. This is the last slide. 3 experiments, 3 sets of experiments, different days. No way. This is still a mystery for us. No conclusion. The obstacle does not seem to work with pedestrian. Here you can see the distributions. This is low competitiveness without obstacle. This is high competitiveness with the obstacle in many different positions. This is one set of experiments. No effect of the obstacle in the distributions of cloggings. The same happens with other experiments with different kind of obstacles, etc. So still, we did not know what happens with the obstacle in pedestrians. Why, in this case, the difference is so big if we compare with the other systems. So thank you very much for your attention. I mean, I can summarize this in just saying that you have this alternation of flow clogging in this discrete flow through bottlenecks. The statistics are very different in the flow intervals and clogging intervals, and this can be used to define a kind of phase diagram that helps to encompass the behavior of many different systems. Some curious behavior, like the faster is slower, but of course still some doubts about what is the role of the obstacle, how is the obstacle affecting to this compatible and compatible load. And of course, it would be nice to see what happens with this kind of behavior that passes through bottlenecks, geometrical constrictions in micro scale or nano scale. Perhaps you are interested in some kind of research with this. Well, thank you very much for your attention. Perhaps I can stop sharing my,
Thank you very much, Iker, for this beautiful and very interesting and appealing presentation. So for the questions, we have now a little bit less than 10 minutes because you took 2 or 3 minutes more than expected. Sorry. Anyway, we are going to organize the questions as follows. If you have any question, please write it in the chat. I will give you order according as we receive your questions in the chat. And please write also your name and affiliation. So there is a first question. There is a first question by Miguel Ruiz Garcia from UPenn, UPM, who says that whether you see any crowd size effect with pedestrians. As there are less people remaining to cross the door, would the exponent increase?
Sorry, I was reading the last one.
Yeah, so he's asking if you see size effects or crowding size effects with pedestrians. As there are
it's a very good question. Okay, Miguel, very nice question. This is a very important thing. Indeed, the first experiments we did with 100 students, and we realized that there was a transition, a transient at the beginning and a transient at the end, 30, 40 people. So the analysis was very limited. Then we moved to experiments with 200 military people, because we need 200 people to at least have some insight. And of course, at the end, there is a size effect. Yes, there is a size effect. We have to reduce the analysis at a given amount of time of evacuation to be in kind of stationary situation. That's a very good question. That's why the silo is good, because I mean you can put the grains and re-put the grains. But yeah, you are right. I mean there are size effects in all these systems.
So if you want, Miguel, if you want to rebate something, or you can use the audio. If you want to rebate or say something, or
okay, no, it was great. Thank you very much for the answer. I would be curious also, maybe for later, if you saw any difference between a kind of people and so on, but maybe you can tell later. Thank you.
Okay, yeah, well, the kind of people, we have not studied these social things yet. Yeah. I don't know if we will study this in the future.
Yeah, or maybe you could.
We try to give very good orders and say, okay,
yeah,
do this, and kind of using people as they were automata or something.
Yeah, or what if you blindfolded them or something that they couldn't see or something? But just curiosity. Okay, thank you very much.
Welcome.
Okay, so the next question is by Martin Lentz. He asked from Paris-Saclay, CNRS, and he asked about the diagrams that you show that suggest that for a large load and a smaller vibration, the critical outlet size may become very large, and perhaps it may diverge. Can this be used to extract a simple continuum understanding of the clocking transition?
Martin, that question is very important, and think that it's not solved yet. I mean, the limit case of very tiny vibrations is no vibrations, which is the silo. And if I can share the screen, let me go here again for this static silo. Okay, this problem, this transition. You are talking about this transition because when you have no vibration at all, what happens is that if there is a clock, the clock is not resumed. I mean, you will never have, if the clock is formed, you will never have, you are here. But the question is, in this static silo, which has not this intermittent part, this transition from no clock to clock is very difficult to analyze. We have been doing that at the beginning. And it's very difficult because you never know if you are waiting enough time to see if there is a clock. Of course, if there is this clock, the clock will rest forever. Okay, so there will be a very sudden transition from clogging to flowing, from 0 to 1. And this happens when you extrapolate to these tiny vibrations, a lot of load, and the discussion if there is a critical outlet size in this static silo is still open, because there will be a very abrupt transition from 0 to 1. And this is still open. And I don't think it will be solved, because we have been waiting for a couple of days with the same avalanche. We have been measuring in silos avalanches with the average is 10 hours, and still not close enough to the hypothetical critical diameter. So very nice question. I don't know how to solve it.
Okay.
If I can just follow up for 1 second. So what's the largest critical aperture size that you observed with confidence?
For inert grains, 5. This is very abrupt. I mean, for 5, no clocks at all. For 4.8, there is still clocks, 10 hours average.
Okay, so something around 5.
Yeah.
Thanks.
The time of lifetime of our human lifetime. Yeah.
Okay, so we have time for one more question, briefly, 2 more minutes, from Juan Aragonés from the Universidad Autónoma de Madrid. So he asked whether, have you looked at the order on the clock side, the time scale for the reorganization this side, is that different from that in the flow clock dynamics?
The order of the clock side. What do you mean, the order of the clock side? Ah, the order in the arrangement. Yeah, that's a very good question. You mean the arrangement of the bodies, or the bodies near the clock, right?
Yes. Yes.
Okay. No. We are on the way. We have been doing some work on analyzing the evolution of the arch. So looking at the beats of the arch with grains. And also we are trying with the simulations of colloids to analyze how the evolution of the arch, the shape of the arch evolves. And doing that, we have observed something that we expected. That is that the system near the arch also is reallocating, is moving, is kind of changing the configurations. But we have not analyzed that. It's a very interesting question. Yeah, we should do that.
Okay, so I think we are a bit out of time now. There is one more question, but I think it would be better if it is asked after the session during the pause. Unfortunately, we have to move forward with the agenda. So I suggest to go to to move to the next speaker. So the next speaker is Yuri Chubak from the University of Vienna, and he will talk about activity-driven topological glass. So whenever you are ready, Yuri, we are happy to hear you.
Okay, can you see my slides right now?
Yeah.
Yes.
Okay. So thank you for this introduction. So the topic of my today's talk is activity-driven topological glass. But before moving to details, I would like to briefly explain the biological motivation behind it. So we're interested in the problem of chromatin organization in the cell nucleus. So there are about a few meters of DNA which is wrapped up on histones and then densely packed into a cell nucleus of eukaryotes. And apparently there are different mechanisms that govern its organization at different length scales. So at the largest length scale, which is the size of the whole nucleus.
Excuse me, Yuri, can you speak a bit louder, just be louder?
Oh, I'm sorry.
Yeah.
Okay.
Bit louder, just, yeah.
All right. So at the scale of the whole cell, each chromosome has a well-defined structure. So it has a compact structure, and it's located compactly, and it is within a compact territory. But moreover, there are compartments that are actively transcribed, are phase separated from those that are inactive. So from those that are silent. If we move down the scale to say around 100 nanometers, the chromatin has loops which are created by a special molecular motor, cohesin, and moreover, there are also action of other molecular motors that constantly inject and dissipate energy at these scales. So what we're interested in is what is the effect of the topology, which creates these loops together with uncrossability of polymer chains and activity on the structure of chromatin at these different length scales. And as a very simple model for that, we use a dense system of ring polymers, which on its own has a series of very interesting properties. For example, in contrast to simple linear chains, each ring polymer has a very compact structure, meaning that it has contacts with only a few of its neighbors. But although it has this very compact structure, there are a special type of constraints that can arise in these systems. And these are the so-called threading constraints, which you can see on this picture on the right-hand side, when one ring is sort of piercing through the opening of another ring. A very important feature of these constraints is that they actually significantly slow down the dynamics of rings in the system. But moreover, they become more pronounced if you increase the length of the rings in the system. And it has been conjectured that based on these two observations, so that the rings significantly slow down the dynamics and the fact that they become more pronounced with increasing the ring length, the dynamics can become so correlated that basically the system will arrive at a glassy state built up solely on these topological constraints at temperature which are much higher than the typical glass transition temperatures. So this conjectured topological glass is formed solely on these topological threading constraints. However, it has not been yet explicitly shown to exist in simulations on experiments because the conjectured critical length for this transition is simply too high to be simulated or produced experimentally. So what we have done is that we coupled this ring topology with activity. We actually have a very, very simple model for activity where we just pick a consecutive segment on a ring. So we start with an equilibrium system, so with fully equilibrated ring polymer melt, and then we turn on activity on a consecutive segment on a ring. And the activity is simply modeled by coupling these active monomers to a heat bath, which has a higher temperature than the rest of the system. So these active monomers here are pictured with the orange color, whereas the passive ones are gray. And note that all the interactions between the orange and the gray ones remain the same, so they're purely repulsive. So the interactions are purely repulsive between all monomers in the system. So the activity level that we've chosen is that the hot temperature is 3 times higher than the cold temperature, but due to the heat flow in these Langevin dynamic simulations, the effective temperatures that we actually measure from the kinetic energy of monomers is lower. So actually the effective temperature ratio is about 1.7, so it's like below 2. Yeah, and the presence of these active monomers significantly affects the dynamics of a single chain, which you can see on this short video. So what is happening is that this active segment is essentially driving the dynamics of the ring in this system. So what is going on is that this hot segment is pulling the rest of the chain and drives it through the mesh of topological constraints formed by other chains. This happens because of the two main facts. The first of all, obviously, the diffusivity of these hot monomers are higher as compared to the rest of the chain because they are coupled to a hotter heat bath. But from the other side, there also exists phase separation between hot and cold monomers. And actually, the phase separation triggers a more dilute environment around these hot segments that also enhances their diffusivity. So what we find in the steady state is that the size of those active rings increases significantly. So they become about 2 times higher than those in equilibrium. And moreover, their structure is completely different. So they have these double folded structures with the active segment usually located at the tip, one of the tips of these rings, which you can see on these snapshots. But moreover, the system also arrives at a partially segregated state where the active monomers are feature more contacts with the active monomers. So the system is partially phase segregated. But more importantly, due to this expansion of rings, their dynamics significantly slows down, and eventually it becomes very, very, very slow, which you can see from the mean square displacements on the left-hand side. And in the steady states, the rings practically do not move. The only motion that is generating is the occasional pulling of these hot segments around. But the whole ring is mainly fixed due to the numerous topological constraints that are formed in the system. Also, the mean square displacements, the centers of the ring centers of mass, as well as the distribution of these displacements, is non-Gaussian, highlighting a glassy state that has been formed. So this glassy state is built on threadings, on these special types of topological constraints that exist in the systems of rings. To analyze these threadings, we employ the so-called mini-pole surface approach, where for each ring we build a minimal surface. So with the help of this surface, we can compute the penetrations between one ring's contour and the other ring's minimal surface. Using this approach, we can extract a few basic quantities that can quantify the threading between rings in the system. So the most important ones are how many of other rings one ring threads and how deep this threading is. And we find that after the onset of activity, the number of threading networks increases significantly. So in equilibrium for this particular system, it's around 7 or 8, and after the activity quench, it becomes something like 20, so about 3 times higher. And in the same fashion, increases the depth of threadings in the system. But besides the generic increase in the depth and the number of threadings, we also observe special types of constraints that form the so-called reef knots, which you can see on a schematically pictured on the right-hand side here. So in this case, you can, these are the so-called reef knots, and just in this case, 2 rings are not linked to each other, as you can see from this schematic picture. There are just 2 rings forming this special type of constraints, and if the rings are pulling in 2 slightly different directions, the state cannot be relaxed at all, as long as the activity is turned on. And on this short video, you can actually see how this happens for a cluster of 4 rings that were initially in equilibrium and building a very sort of deep cluster of threadings. Once the activity is turned on, these hot segments start to expand in the system, but each of them forms a so-called reef knot with each other. So what is effectively happening is that they are connected to each other in this particular way, and they cannot diffuse at all because they are bound to each other with this reef knot. And apparently, this phenomenon is driven by the non-equilibrium phase segregation. What we have done additionally is that we have simulated similar systems of ring, but with different length, by keeping the fraction of these active blocks the same. So what we find is that for those systems that have a tendency toward phase segregation, which is quantified by means of this order parameter phi, which simply quantifies the number of like contacts between particles, we find for the 2 bigger systems that we simulated a tendency towards phase segregation. However, in this case, the systems also arrive at the glassy state with a very pronounced threading. On the other hand, in the smallest system with the rings of length 100, which also have like which have 1/8 of their monomers active and coupled to the same temperature, we don't find a tendency towards phase segregation. What we find is that the dynamics of the system is effectively equilibrium-like, and that the center of mass moves effectively with the temperature, which is simply a weighted fraction of the temperatures of all monomers. So in conclusion, the combination between topological constraints and activity can lead to interesting states of matter. For example, in this case, the combination between activity and topology creates the state of topological glass by deforming the equilibrium ring melt and promoting the interring threading. So the observed behavior has certain similarities with the phenomena in chromatin that occur at these smaller scales when we observe the formation of loops through these loop extrusion mechanisms. A few of them are the directed motion of highly transcribed genes, as well as the expansion of these long transcribed genes in size. Furthermore, there are these phenomena of highly correlated heterogeneous dynamics of chromatin at these line scales, as well as the segregation between active, passive chromatry. So perhaps this phenomenon might be important in explaining some of these phenomena, or can have a certain contribution to the understanding of them. Yeah, so I would like to thank my collaborators from the University of Vienna, so Jansberg and Christos Likos, as well as the collaborators from the Max Planck Institute for Polymer Research, Stander Patrick and Kurt Kramer. And thank you for listening to me.
Okay, so thank you very much, Yuri, for this very interesting presentation. So now we have, in principle, we have 5 minutes of questions. I remember you to write them in the chat. Right now, there is no question I can see. Maybe just to break the ice and ask you something. So can you tell us, Yuri, please, will this phenomena be relevant, for example, beyond chromatin organization, for example, in the problem of polymer folding, like, for example, protein folding, RNA folding? Do you think that this sort of behavior can contribute to some of the mechanisms like trapping dynamics during molecular folding?
Well, actually, I haven't thought about it, but well, it might be possible if the topology of the polymer plays some role for that, but probably for the protein folding, it won't be important, I would say. So, because what is, I guess, most important for the protein folding are the different types of interaction between the protein beads. Whereas here, this phenomenon arises on the basis of this particular ring topology and activity that acts locally on some segments. I
see. Okay. Okay, thank you. So there is one question by Martin Lentz from Paris-Saclay, CNRS, and he asks, is it known whether coenzine forms the same kind of hot domains that are present in your model? Do you know whether the behavior system is similar if multiple hot zones are present?
Well, I think it's not entirely clear whether cohesin is responsible for this behavior in vivo, or it has been shown that it creates loop in some in vitro experiments. I think it was done a few years ago, or yeah. So whether it causes phase segregation, it's probably not entirely true that cohesin alone through this loop extrusion causes the phase segregation between active and inactive chromatin parts. There is certainly some combination between chemical phase separation and the active phase separation. And the active phase separation usually makes the chemical phase separation less pronounced. So there is always, in vivo, there is always a combination between these two effects. So the combination, so the phase separation through different interactions between these chromatin beads and through some active processes that are on top of it.
Okay. Okay, thank you. If you want to rebate, Martin, or any other comment by...
Second half of the question about are your conclusions changed if instead of having one hot domain you have many along the chain, right? Which could
yeah, this is very interesting. So in this case, we had this very simplest model when there is just one segment. But in reality, if there are multiple domains that are expressed in a real chromatin, right? So We don't know. So the question is, we don't know. We have not tried yet, but certainly to have some effect. It might enhance it if these domains are located maybe at different tips of this ring, or it might not cause it at all. So we'll probably try it at some point, but we don't know yet.
Okay, thanks.
Thank you.
Okay, so if there are no more questions or comments, we are very good in time, I think. We move forward. And therefore we go to our next speaker, who is Julian Simpson from TU Dresden, who will talk about apparent phototaxis enabled by Brownian motion.
Okay, thank you.
Whenever you want.
Let me share my screen. So can everybody see my screen?
Yeah,
Good.
fine.
Okay, so I just realized I didn't have an acknowledgement slide, so I thank our funders, the Volkswagen Foundation first, and obviously all of my group and my collaborators. And today I want to talk about understanding phototaxes. And one of the reasons why everybody is so interested in active matter is because active matter research promises that it can help us understand behaviors and things that happen in nature by simply forming more simple models. So the question for us was, if we can move towards understanding a complex phenomenon like phototaxes that we can see in different types of bacteria by using our photocatalytic micromotors. And today we want to fill in like the missing puzzles in there, at least for one of our systems. But let's first briefly have a look at an example of biological phototaxes. So what we see here is an SEM picture of Halobacter salinarum, which is a phototroph bacterium that can actually perform a certain type of phototaxis. I say a certain type because in biology there are many different functions and many different types of phototaxis. So here you see a video where Halobacter salinarum switches from this forward motion by being exposed to a blue light pulse, and it directly moves backwards. So this little video is from MPG Magdeburg. So acknowledgement goes to Professor Marwan, who was playing around a few years ago with these little bacteria. And so if we want to have a similar effect on our phototactic, on our particles, we first have to find a way how to make active particles react to light. And one very good way is actually having the phototactic the active particles gaining their energy from light, which is very similar to what phototroph organisms do. And to do so, we need to find a certain type of material that can actually interact with light. And a good option for this are semiconductors. And in this work, we went for titanium dioxide, which is here, the fourth in this diagram. It has a band gap of about 3 electron volts. And what happens with the semiconductor, if you shine light on it, I can show it to you here in principle. So we have the semiconductor, the particle, and once we switch on the light of the corresponding wavelength, which would be UV light for this band gap of about 3 electron volts, what happens is that the electrons that normally are here in the lower valence band, they gain energy, they take up the energy from the light, so they're promoted to the conduction band. That means in the valence band, we have here this hole, and we have the motile electron in the conduction band. And these both charge carriers are able to catalyze chemical reactions. This is called photocatalysis. Here, the example is water, because it's what we always object for, like what we always aim for, but it obviously also happens in peroxide. So it happens with different photodegradable fuels. Obviously now you might tell me like, hey, what you're showing us is an isotropic particle, this is not going to move, and you're perfectly right with this. So my student Linden started making Janus particles out of them simply by taking these tiny little colloids, putting them into a monolayer, which you see here schematically, and then by depositing a metal layer on top. So this metal layer can be anything. In this case now it's gold. And here we see, just shift this movie. Here we see the advantages of these particles. First of all, compared to normal platinum swimmers, you see they're really, really, really fast. Oh, you see that they're really, really fast. Second advantage is that we can stop the motion. So you see we have a really precise on and off switch. And now that we start it again, you can also see that there are a lot of curious and interesting interactions between these particles. So they have kind of a social life, but we'll leave this for another lecture. It would be too much for today. You see, these particles already have a lot of properties, but we still wanted to add more features to play around with them. So what Lentz did was adding a nickel layer below this catalytic gold layer, just simply to be able to direct their motion by a magnetic field. However, what we got was kind of unexpected, because when additionally to this nickel layer, we also limited the light to a certain stripe. What you can see here is that these particles, without changing the magnetic field at all, they move back and forth, right? So this is really intriguing, because we didn't add an additional stimuli. We just had a permanent magnetic field there, and the particles did something that really reminded us of the Salobacter salinarum, because they move back and forth. So in order to understand where this behavior actually comes from, I'll give you a little more in detailed overview of the setup. So we have a magnet, which you see here just in scheme. So the magnetic field is constant over the whole setup, and it makes the in-plane magnetized nickel layer here orient in the way that you see schematically here. Then what we see here is that everything, like the whole point of view field, is irradiated with white light, but additionally we have this stripe of UV light which gives activity to the particles. A little trick that we played here was adding green light, because our camera wouldn't see the UV light, so that you can see this irradiated stripe here. We added the green light. And what is happening when our particles, when the UV light is on and our particles get active, is that they do these back and forward motion, which is not really obvious to see. So we started looking at different influences, and at this point, our student Lucas took over this project. So in order to manipulate the magnetic field a little bit more precisely, he created like this little coil setup. So we were really sure we had a homogeneous setup. We could orient the field differently by having implementing here this little turnable overview, but neither the field strength nor the orientation of the field changed much in its behavior. We could change the nickel layer thickness. Also, the phenomenon showed pretty robustly, and we changed the peroxide concentration, the light intensity, so that we obtained different swimming speeds, but still the phenomenon kept showing. So it's a really robust phenomenon, which I'll just repeat for you now. The particles move out of the stripe, they go into the dark area where they're not really active anymore, turn back and move back to the other direction. So they have pretty much a stable orientation. And when we're thinking where the stable orientation comes from, we remembered our old work with the guys from Stuttgart. So we contacted them again, and we were looking a little bit more in depth here. So you see that B, like the magnetic field, fixes the orientation in that plane, so the particle can't move in this direction. And this angle theta here, this is what we found already in 2016 in this old paper, that the angle theta of active particles is usually quite stable, and it's stabilized because we have this interaction of the hydrodynamic interaction with the wall, the gravity and the chemical field, and all these lead to a certain balance of the particle once the particle is active. So when we looked over our experimental field of view, what we got is three sections, right? We got a dark area, which is blue here. We got this little bit of a gradient area where we have less intensity, which is green. And we got this full light intensity here in the middle, which is yellow. And what we can see is when we describe this by this phase plot, in the yellow area, we get this really strong red attractor here, so that the particle is always oriented in this direction. So when the particle swims through the irradiated area, it has basically no option to turn anywhere, because in this orientation, it's fixed by the magnetic field, and in this orientation, it's fixed by its own activity, by this red attractor here. Once we get to this area, which is here green, where the light intensity declines a little bit, what we see is that the attractor is still there, but it's destabilized by the settle point. So whatever wants to destabilize the particle there is probably more likely to succeed than in this yellow area. And then when we look at the blue area, where our particle is basically not active, then there's also no precise orientation of the particle given. So Luca summarized this super nicely in this little video here. When the magnetic field is on, which we see here blue, the particle is free to rotate in this orientation, but the yellow cap is always in this 90-degree angle to us. Once the activity is on, the particle is also oriented here, so it will move forward. But what we've seen is that when the activity is off, we can kind of destabilize the particle. So the question was, what is it that is destabilizing the particle orientation and reorientating it, flipping it back into the light area? And our suspicion was that it's actually just Brownian motion, very simple Brownian motion. And in order to test this hypothesis, that basically once we have here, so once we're in this area of irradiation, we have everything fixed. But once we're in the dark area or in the transition area, Brownian motion can kick in. So we see in the yellow part, which is we have this ballistic motion, and here it's basically just Brownian motion, no ballistic. So in order to test this, what we did was we produced particles in different sizes. All of them were motile, and the behavior kept showing quite robustly. We could observe that for the smallest particles, we had a bit more of flipping. So the Brownian motion that kicked the particles around and reversed the direction was actually sometimes even kicking in the illuminated stripe. When we had these intermediate sizes, 1 and 1.2 and 1.6 micrometers, the particle behavior of moving back and forth was very robust. So the stabilization was very strong here in this active field, but as soon as the particles left this irradiated area, they had the freedom to be flipped around by the Brownian motion. And if we look at this part here, where we have our 3.5 micrometer particles, here the Brownian motion is actually not strong enough to flip the particle efficiently around, so that this behavior almost never shows. So we see that the rotation probability, or that is caused by the Brownian motion, the effect of Brownian motion on the particle, actually enables this flipping around particle, and we get here an area where this is optimized. So to conclude, we have this light-driven magnetically capped colloid, which shows this kind of scotophobic behavior. So it seems it's afraid of the darkness, but if we put everything together, that it has an active sliding position, which is stabilized in the irradiated area, and the stabilization is missing once it reaches to the dark area where Brownian motion can flip it around. It's all basically very simple physics. It's an amazing combination of simple physics. However, this is so easy that it's probably not really applicable for bacteria, because the mechanisms that lead to phototaxes in bacteria are usually cascades of enzymes reacting and gradients and formations. So that what is here found for these simple active particles is probably not one-to-one transmittable to biological systems. So with that, I have already reached the end. And I would like to thank you for your time and attention, and I'll take any questions.
Okay. Thank you very much, Julian. So there are actually 2 questions already in the chat. The first question is by Miguel Ruiz Garcia from Penn University and UPM, who asked whether the flows of the fluid around the particle could contribute to the turning around in the inactive region.
So if we go back, let me see if I can go back. If we go back here, most of these flows around the active particle are caused by the activities. So in the inactive region, there are not so many flows. What is dominating there is this torque of the gravity, or the force of the gravity, which orients the particle bottom heavy down. And there, if it's actually being it's always undergoing Brownian motion, right? If you look at passive particles that are bottom heavy, they always do this kind of shaky Brownian motion, where you can see that they get reoriented a little bit. And once so there are not so many flows, but there is really Brownian motion that turns it around, and then it becomes a little bit active anymore, because it enters this green area where there's a little bit of light, and then it can just move back. So I think it's rather the absence of these flows than the flows that turn it around.
There is also the question by Martin about, which is related to this one, which is maybe whether changing the shape of the particles a bit spherical, or maybe change their shape by sticking a smaller bit to them, you could directly look at how and when they turn. I mean,
yeah, we would have to make the particles quite a bit larger, I guess. So our particles are about a micron. So we do have, for example, ellipsoidal particles, but it's really hard to see slight changes of orientation in an optical microscope. What we could probably try is an approach similar to what Per Fischer's group has shown, that if 2 particles just couple,
okay,
that might be an option, yeah.
Okay.
Thank you.
So let me ask you briefly another question, first question. So all these experiments are done in a solvent of distilled water, is that correct?
Yeah, we usually use mini-Q water, and we add a little bit of hydrogen peroxide. So these experiments are done in 0.5% of hydrogen peroxide, simply because it increases the speed of the particles massively than if you do them in just water.
See. So do you expect, if using, for example, changing the pH of the solvent, putting more protons in the solvent, could modify the reaction kinetics in the particles, these induced, light-induced reactions? Do you think by using a more acidic solvent or basic solvent, could you change the kinetics of these reactions, and therefore the activity of the particles?
So what we have seen is that if we change the pH of the reactions, it depends on how much we actually change it, right? What you also change is the ionic content of the solution. So, and if you have too many ions in the solution, your particles will just stick to the substrate. But there is, I suppose there's an interplay where your whole solution is like positively charged enough, but does not yet stick. So that your Debye layer is too much depressed. There, it could be that the particles actually move to the opposite direction.
I'm not talking about ionic solutions. I'm talking, so I'm not talking about ionic strength, but pH, protonation.
Yeah, but I mean, obviously, if you protonate your solution, you increase your ionic strength, right? So at some point, your particles will stick. We have tested this for a different type of particles with bismuth vanidates, where about pH 2.5, the particles really stick. But we definitely see that changing the pH changes drastically the properties and also the fluid flows.
I see.
So we've not tested this for these titanium dioxide particles, but we have an article on bismuth vanidates, where we actually see that depending on the pH, the particles move flat or stand up for motility.
I see. Okay. Okay, so thank you very much. I think it's 10:15. So I wish to thank you very much, Julian, for your very nice presentation. So we are in time, and I think it's time for the pause. And as I told you in the beginning, I was not very fond of these Zoom conferences, but I think we did pretty well, and I'm much more happy. So I think it was very well done. I wish to thank all the participants and all the speakers of this active matter session. And it's my time now to pass the word to, I presume, Laura or Juan.
You water, whatever you like. Morning. Welcome to the second part of our first day of our mini-colloquium. My name is Timon Idema. I'm at TU Delft in the Netherlands. In this part of the session, we'll again have one invited talk and two contributed talks. The invited talk is given by Martin Lentz, who you should all be able to see now. Martin and I have known each other for a long time, since we were PhD students, when Martin was in the group of Jean-François Joanny at Institut Curie. After he got his PhD, he went to Chicago, if I remember correctly, for a couple of years, and then landed himself a CNRS position back in France, where he since recently got promoted, I think 2 years ago, to the more senior CNRS position. He leads a theoretical soft matter biophysics group and has clearly very broad interests in various aspects of biophysics, including membranes and cytoskeletal elements, which, as some of you may have heard, I am gratefully using in my classes, which are concurrent with this colloquium. And to illustrate this point, Martin visited us, I think, 2 years ago or so, when he gave a seminar in the afternoon, just talking about persistence length, which we had just discussed in that same class in the morning. And students remarked that they were very happy to see that these things are actually used by active researchers. Also, Martin has many talents. I can attest that he is also a great cook. And a great speaker. I have enjoyed listening to him on several occasions. Today he will talk about entangled networks, and to be precise, it's talk is about how you get elasticity from entanglements in branched actin. How we're going to do is, so the whole talk is about 45, say, a little under 40 minutes, and we have time for questions. So please, if you have a question during or after the talk, especially after the talk, please do indicate in the chat, and we'll experiment a little bit. I'll give you the chance to ask your question live over the audio, but I will moderate. Martin is staring at his screen, I guess, because you're trying to get up your slides.
Yes, let me try to do this. Next, so should be good.
And Martin has asked that a couple of people at least keep their video on, so that he has some people to talk to. I see there are 3 people with video on, but if a couple of you would like to join us in that, you're welcome to. And then Martin, please tell us about your latest research.
All right, thanks. Yes, so what I wanted to tell you about a little bit today is some form of activity that exists inside living cells, and it resides in its cytoskeleton, this network of biopolymers that powers many things inside the eukaryotic cell. And it produces activity in 2 large ways. One of them is the molecular motors that are perhaps more famous. And another one is the fact that this actin polymerizes, and especially it polymerizes into some specialized branched structures. And those are the ones that I'd like to talk to you about today. There's a lot of biochemistry involved, but perhaps I'll convince you that some of the properties of this branched actin are actually more physical than what people realize or typically assume. So here's my rough sketch of where branched actin comes into play inside the cell. This is just one specific example of a cell that's crawling on a glass substrate. You can see the nucleus here. And as it does so, it has to advance its front end, push its membrane forward. And that is done precisely by the structure that I mentioned, branched actin, that forms those filaments that have, as Timon would put it, a certain persistence length that's long enough in order to make them, stiff and pushy in a way. And at the same time that this happens, they branch on the side. There are some specialized proteins that nucleate branches on the side, which push against the membrane. All right, so what kind of mechanical characteristics? I'm sorry about, sorry about this. All right, so what characteristics do you have for those networks? Well, there's an interesting thing that has to do with them, which is the fact that they're quite adaptable mechanically. Depending on the conditions under which you grow them, they might have very different mechanical properties, and that's illustrated by this study. This study here, where people have grown such gels and then compressed them with AFM, either during growth or only after. And you see that if you grow your gel under a very small load, then it's going to end up, so you grow it, and then at the end, you probe it with this test stress. And if you grow it under a small load, it's going to end up being very soft and increasingly stiff if you grow it under higher loads. And there's also direct evidence that this happens in living cells. So where does this adaptability come from? And there's one possible source that is biochemistry, and that's perhaps the one that's most often assumed. Just to give you a little overview or introduction to the kind of biochemistry that I'm talking about here, in branched actin, what you have is typically those filaments that I mentioned before. They are grown by addition of monomers at the end of the filament, but at the same time that this happens, there's branches that proliferate through the addition of those branching proteins that nucleate new filaments. Those guys have to be activated by specialized proteins that are only found at the membrane, but also to prevent the branches to proliferate too much. There's some capping proteins that come and arrest the growth of some of those branches. And all of those are complicated processes, and all of those actually may be mechanosensitive. For instance, people have proposed that the branching protein ARP23 comes in more likely on a filament that's curved. So could it be that, under stress, the filaments have a different shape, or, maybe the stress itself has some influence on some of the mechanisms? And could it be the reason why the networks grown under stress are stiffer than the ones that are grown without stress, which is obviously important for the cell to be able to deal with its environment in a way that's consistent with the constraints that exist around it? Well, that is a possibility, and people have researched it, but there's not extremely convincing evidence that this might be the case. And that leads me to perhaps propose that this versatility, this adaptability of those networks may be due to more physicsy reasons than mechanosensitive proteins. And in order to tackle this subject, I'd like to go in 3 steps. And the first one is to tell you what I think is pretty special about the elasticity of branched actin, and how this specialness might help it be as adaptable as it is. And it's essentially, I'm going to try to convince you that it's a material that's extremely susceptible to outside stresses, more than the biopolymer networks that you're used to. Then I'll try to go a little bit more into the specifics of what the elasticity of those networks look like in the absence of an outside growth force, and then present to you some of the things that we've done trying to explain what the growth force looks like. And I have to apologize, perhaps some of the data that I'm going to show is slightly outdated, but I think the basic idea is still worth conveying, and I hope this is enough for us to have a discussion afterwards. So let me start with the reason why I think branched actin is special. And in order to do this, I'd like to take a step back and discuss a little bit the mechanics of polymer networks, networks of beams, and perhaps the history goes that the ancestor of those is the mechanics of larger scale beams, as were studied, for instance, by Maxwell. So that's during the 19th century, right? And I don't know much about the 19th century, but I like to imagine that the main preoccupation of those people were to build Eiffel Towers and bridges. So in my little inside story, Maxwell thought about what does it take to make a nice bridge. If you have, steel beams, they're of course most resistant when you take a steel beam and you try to stretch it or compress it. It really doesn't do that. And so you should build the stiffness of your bridge around this very strong stretching stiffness of the beams. Of course, the beams are also difficult to bend, but comparatively, it's actually easier to bend them than to stretch them. Now, here's 3 designs of bridges, and they are different in how many beams they are compared to the number of points, what the average coordination of those points is. And you see that if you have a very low coordination, you can imagine, for instance, that at those vertices, there's a hinge that allows your system to bend. Maybe they drilled a hole and put a rivet in there or something. And clearly this guy is not stable. It's just going to, the hinges are going to give, and then the whole thing is going to collapse into the gorge. Now, you could have this alternative design here where the weight of the bridge is now supported by the bending stiffness of this rod here. So it's going to be better, but if you are going to have a heavy load on the bridge, then maybe that's also going to bend and badly. And the best design, of course, is in a situation where there's, be careful about this, there's as many constraints on your system, as many stretching constraints, as there are degrees of freedom. And that is the guarantee that your system is going to be completely stabilized. You can't, if you put a pedestrian here, it's going to push down, that's going to tend to extend those beams and to compress this one. That's, of course, going to be very difficult to do. And that's the reason why the pedestrian is not going to fall down. So I want to outline 3 regimes of elasticity: floppiness, an elasticity where there's enough bending constraints to stabilize your system, and then one above a second critical value of the connectivity where just the very strong stretching constraints are sufficient to stabilize your system. Now, that's actually applicable to biopolymer networks as well. And this is a model of those, a triangular network where you remove a certain fraction of your bonds at random, which gives you an adjustable coordination. If you are above this critical connectivity, then your network is mostly stabilized by stretching and you have a very high elastic modulus. Please mind the logarithmic scale. And if you go below the lower critical connectivity, then your system, the modulus goes to 0 and your system goes completely floppy. And there's this intermediate regime where bending is sufficient to stabilize your network. And presumably that's where most biopolymer networks live, somewhere in the middle of this region here. All right, so now let me go back to the specific example of the branched networks that I presented before. And let me build such a branched network iteratively, right? So I'm going to just pretend that this branched network is a network of vertices and bonds between the vertices. Here's the simplest branch network that you can have. It's not even branched, it's excessively simple. But if I take this simple example, how many degrees of freedom and how many constraints do I have? Well, there's 3 trivial degrees of freedom where you can take this dumbbell and translate it up or sideways or rotate it in a way that is not going to deform it. Now, there's another mode of deformation, which would consist in stretching this bond here. That's an internal deformation. That's what I call 1 internal degree of freedom. Now, how many constraints are there in this system? Well, there's also 1, because this steel beam or biopolymer beam constrains those 2 points to lie at a certain fixed direction of one another. I mean, you can deform it, but it's going to cost you energy. And the other modes of deformation that the other degrees of freedom here are not going to cost you any energy. All right, so that's the simplest case. And now what I'm going to do is that I'm going to add one more point to my filament. What does that do? It adds 2 degrees of freedom, the translations of this point, and 2 constraints, the stretching of this bond, and the fact that bending this hinge costs you energy. There's a persistence length to your biopolymer. Now let me consider a branching event. Now I'm adding what? 4 degrees of freedom for 2 points and 4 constraints, 2 stretching and 2 bending, and so on and so forth. I can add, sorry about this. I can add arbitrarily many branching generations and, you know, an arbitrary topology of branching. And I'm always going to have the result that I have as many constraints in this system as I have degrees of freedom, because I have no loops at the end of the day. And that's the difference with regular polymer networks. Those branch networks have no loops, almost by definition. And I end up having them where? Well, exactly at the place where I have this critical connectivity. And so the prediction from this simple argument, which is, I guess, the way you would usually do biopolymer elasticity, is that those networks should have a modulus that's exactly equal to 0 because they are critically connected. All right, so that's a nice story, but it's a little bit of an issue because we know that it's not the case, right? We know that those are some of the stiffest structures that are found inside the cell. So, you know, where is my sanity? Well, the first thing that I have to say for my defense is that this is a reasoning about the linear response of the network. So the AFM experiments that I proposed to you before are a little bit blunt and they don't have a very high force resolution. So we went to a much more sensitive system that was developed by Olivier Durour and Julien Heuving at ESPCI. And what they have in their system is magnetic colloids that have the shape of cylinders. And when you put many of them in a solution and put a tiny magnetic field, you may know that they tend to organize along a chain of colloids. Now you can be slightly more clever than this and treat the surface of those colloids with the ARB23 activator, which means that as you introduce actin and all the necessary ingredients, you're going to have a preferential polymerization of a branch network between colloids. So what I'm going to do first is I'm going to align those colloids with a very small magnetic field. So don't mind the stresses that are induced by the interaction between colloids. I'm going to let the network grow for a little while. And then I'm going to probe the mechanics of the gel by hiking the magnetic field and therefore the attraction between the colloids up and then taking it down. And I'm going to look here at the relationship. So I'm going to get a stress-strain relationship from which I can derive a modulus versus stress relationship. And lo and behold, if I go to smaller and smaller compressive stresses, it turns out that I never hit a plateau. It looks like the modulus is, to our resolution, as small as you want it to be, which suggests that indeed there is no stiffness in the linear response regime. This material is special in that it has a nonlinear response for arbitrarily small compression, which is consistent with the argument that I gave you before. Now, this might seem a little bit strange because, at least as physicists perhaps, we're used to what I might call usual materials, where you have a certain finite modulus at small stress. And then as you go up and up in stress, maybe you have a change of the modulus. Maybe there's a stiffening in this network or something like this. But there's actually another community that knows things that work in the same way as what I've described, right? So this is, again, data on the branched actin. And now let me superimpose on the same graph, without any adjustable parameter, data for other materials. What are those materials? Well, you have a list here. You have sheep's wool, human hair, you know, anything you can, many things that that are not directly related to the to actin, but the commonality between all those materials is actually that they are materials that are extremely under-coordinated. They are threads that are entangled together, and that's, you know, if you just count the number of constraints and degrees of freedom of just a thread or, you know, a ball of wool, then you would never expect it to be rigid, and so it responds in this way. Okay, so, you know, that's just a picture for now. What should I draw from it? And this is what I'm going to try to explore in this second part, where I try to understand the elasticity of those networks in the simpler situation where there's not a force under which they are grown. That's work that was done by a postdoc in my group, Mehdi Bouzid, as well as Cesar Valenza-Gallardo, who was doing the experiments that I've described before. And the basic idea here is that the picture that I've presented to you is too simple. I've told you, you know, I have a tree that grows from here and up and up and up and up, and it becomes larger and larger. And if I push on it, its stiffness is essentially 0. Why was that? Well, let me give you an alternative explanation than that talk of constraints and degrees of freedom. And the idea here is that as I push on this tree, the force that I'm exerting is integrated over a very large surface. And that force is focused down on the one filament that I have at the root. So large force, small spring constant, essentially I get an arbitrarily large displacement, which is the same as having a 0 modulus. But that picture, which is equivalent to what I said before, is too simple. Because I have such a tree, what's going to happen as I push on it is that it's going to bend to the side, and it's going to inevitably run into the next neighbor, into the next tree over. I'm going to create a contact, or, you know, maybe entanglement is not the right term, so we can discuss terminology if you want. And that's going to add some extra constraints in the system. And by adding those constraints dynamically, you will stiffen your system as you go. And we tried to test this idea through numerical simulations, where we made networks that are initially completely no contacts between the filaments. They have this branch topology and the same anisotropy that biological networks have. And the basic rules of this simulation is that, you know, let me take a couple of vertices along this filament. It's very expensive to stretch a link. A bond is actually, sorry, a hinge is relatively easy to bend. That's essentially my unit energy in my system. And I also make my vertices impenetrable in such a way that, you know, if I have two filaments that run into each other, I do have an extra constraint in the system. And here, what you can do with simulations is verify what I was suggesting, that the contacts are very important, because you can turn the interaction between filaments on and off, and you can immediately see that if you turn on the interaction between filaments, you have a much larger modulus, and you essentially have a lot of stiffening, which, you know, confirms that this is an extremely important part of the mechanics of those under-coordinated networks. All right, so what do we understand out of this? Well, it turns out that the source of our understanding is a pretty funny one. It's a guy in the 1940s who was working at the Textile Institute of Johannesburg and was interested in sheep's wool. And he has developed a pretty neat mean field understanding of what made its wool rigid, which essentially goes as follows. Imagine that you have a box with a long filament of wool in it. And you just start by imagining that your filament it has a certain thickness, but it's a completely phantom filament. It can go over itself in, you know, a completely random fashion. And what you do is that if my filament is phantom, you count just by chance how many overlap I'm going to have between two filaments. And you say, in my more realistic system, this is where my contacts happen. And that gives you a certain density of contacts. Of course, that depends on the volume of your box. If you make your box smaller, then you're going to have more of those contacts, essentially because there's just less space in your system. And then you say, the response of my system is just the number of segments of this kind that I have between two contacts, and then, you know, multiplied by the mechanical characteristics of bending one of those little pieces of segment. And then at the end of the day, you end up with a certain pressure volume relationship, which goes that P equals 1 over V3 that you see displayed here. All right, let's try it. And here you go. that's the comparison between the stress versus volume to the -3 in both the simulation and experiments. And I think the agreement is encouraging, and it actually turns out that we've become more encouraged with time and more data that I won't be showing today, unfortunately. So that's nice. Now, what I'd like to do is try to harness this understanding of the networks that were growing without stress, and then understand how the active growth against the loads modify those characteristics and makes for that very adaptable mechanics of my networks. So let me go back to our favorite experiment. What you can do now is turn on the magnetic field earlier than I have before, and make it so that this magnetic field is on during the growth. That's represented by this blue line here. And then at the end of the growth, you also probe it. And it does appear that when you do this, your network acquires a certain plateau elastic modulus. And that plateau elastic modulus is a function of how much you've pressed on your system. So how do I understand this? Well, let me try to reproduce this in the simulation first. It's a little bit involved to really make a growth simulation. So I'm going to do something simpler. What I'm going to do is that I'm going to start again with a ghost filament. I'm going to compress it. And as I compress it, my filaments are going to go through each other and not each other with their neighbors and become entangled. Then I'm going to unghost my filaments, if you will, reactivate the interfilament interactions and relax it. But now those knots are frozen into the system and they can't relax. And then I have a structure that's now pre-stressed because those filaments are pushing and pulling against each other. And when I do this in the simulation, and again, I apologize for the slightly outdated data that I'm presenting here, it does appear that you're building up more and more of a plateau as your system becomes more and more pre-strained. Another important element of understanding is trying to relate the stiffness of your network, because that's basically my explanation. Your network is as stiff as there are contacts within it. And that's the great interest of the simulation, that you can count the contacts in the unstressed and unstrained and pre-strained networks and see if you have a correlation between the number of contacts that you have there and the stiffness that you're going to get. And it looks at least promising here. And again, it's our understanding has changed a little bit, but we have data that I think is more convincing than this now. All right, so now I'm drawing this parallel between growth under force and having pre-strained and built-in entanglements in the system. How can I relate the two? And the answer is we try to relate it through a simple scaling theory. And the basic idea is to say, well, you have a certain distance between your filaments. That's a network that's pretty entangled, and then you have filaments coming out of it. Now, what happens if your network is grown under a pushing force? That's the force here, right? Well, what's going to happen is that the filaments that you have at the top of your network, the ones that are currently actively growing, instead of growing straight, they're going to grow in some bent fashion, right? And so as they grow, they're going to knit themselves into the other filaments that are already there. And this is where your entanglements are going to come from. Not from an overall ghost compression as in my previous protocol, but through the dynamic growth and weaving of your different filaments. And so if I assume that I have a certain length of filaments between two entanglements, so that's the length of the filaments between the point where it was last entangled, right? And then it grows and it grows. As it becomes longer and longer, it becomes more and more deformable. So you have a larger and larger deformation U. And my scaling argument is to say, when U becomes so large that it's going to hit the next filament over, well, that's where the next entanglement is going to come in, right? So you just have to cook this a little bit self-consistently. And at the end of the day, you predict that your initial network grown under a pressure PG is going to have a certain finite density of contacts within it. And because it has a finite density of contacts, just like in the sheep wool's case, it's going to have a finite rigidity to start with. And again, that's just a preliminary picture. Maybe it's just an illustration of the way we want to demonstrate this. If you feed this theory and you just plot this theory, right, saying what's the K0, which is the modulus that was going in front of the sorry, so that's the plateau modulus that you get in the beginning. If you have a system that grows under very small stress, then you have a certain plateau modulus that is very low, that was not detectable before, but it just comes from the finite density of your network and the random encounters between the filaments. And as you increase the growth stress, you should get more and more frequent entanglements with this specific power law in the growth stress. And what I'm showing here is the experimental data. It's not averaged, and we have an issue with this thing. So that's not yet very strong support for what we're saying, but we have a good idea of why those points are off. And we're actually hopeful that this will become more convincing with time. So that's essentially what I wanted to tell you about, right? Just a little reminder of the substance of what I said. What I said is that you have to be careful when you think about branched actin, because it doesn't quite work in the same way than the other biopolymer networks that you're used to, right? It's more like sheep's wool than it is as cross-linked actin, right? Which is very counterintuitive from a point of view of biochemistry, but it's really the topology of the branched network that makes that true. And standard theories of biopolymer elasticity would suggest that branched actin should not have rigidity, but it's essentially what I'm proposing is that the entanglements that built in as you compress the network and as you grow it under force act as dynamic crosslinks that add constraint to your system and finally endow it with a certain mechanical stiffness. And that makes for a self-organized machinery that helps the cell adapt the stiffness of their branch networks to external conditions under which they grow. And perhaps going outside of the strict confines of branch networks, that discussion can be an inspiration for us to think about other networks, also cross-link networks, or at least ones that are not too dense and not too stiff, because there's one effect that's typically not discussed in the study of those systems, which is as I compress my system more and more, what about the places where the filaments run into each other, not at crosslinks, but in between crosslinks? And it could be that that stiffening could also play a role in other networks, even though I don't have direct evidence of this just yet. So that's about what I wanted to say, and I thank you very much.
Thank you very much, Martin. Thanks also for sticking to the time, despite my mistakenly saying that you had 40 minutes, you had 30 minutes, right? But you stuck very nicely to it, which also means that we have some room for questions. If you have a question, please indicate in the chat that you have one, or virtually raise your hand, and I will give you the option to ask your question yourself. And I see we have here one from Miguel Garcia.
Yeah, hello, thank you. So I had a question about how important are fluctuations like thermal or active fluctuations in these systems?
All right, so let me start with active, and I think there's one important element that those networks typically don't have much myosin in them. So there's many sources of activity, and you can never get rid of it, but that's typically the largest source of activity in the cytoskeleton, and it's not very present in the system. Moreover, in the reconstituted system that I showed you, the activity is essentially just present at the growing end of the filaments, so just at the surface of the network, and comes from the polymerization, which induces the entanglements, but you don't really have bulk activity. Now let me go to the more thermal side of things, and indeed, when we discuss the rigidity of actin networks, for instance, we're used to thinking about thermally fluctuating filaments. That's a slight bias that's actually induced by the fact that artificially produced actin networks typically have a pretty large mesh size. often microns or more. And those networks have a distance between branches that it's much, much smaller. So essentially, if you, right, so we're way under the persistence length of the filaments. And so, even though thermal fluctuations might have an influence, it stands to reason to say that it should be pretty small compared to that of standard actin networks. And indeed, it seems that we can, account for many of the things that we see without having to introduce that.
Okay. Yeah, it makes a lot of sense. Thank you.
Thank you. So we have another question from Anton Suslov.
Thanks. My question was about the branching factor. How important is it that there's only sort of a twofold branch in actin? If you had more filaments coming out, would that significantly change, say, the stiffness?
So main filament here. Make sure I understand your question, right? So the main filament here, and then one branch from the left, and one branch on the right.
Yes, for example, yeah.
So we haven't done simulations with this, but I can tell you about the constraint counting arguments that I've presented to you, and that would stay the same.
Right, great.
Yes. Does that make sense, or?
Yeah, that makes a lot of sense. Thanks.
So it's really the absence of loops that makes this counting argument tick.
If there is room for other questions from people present.
Yeah, I have another question. I guess that there is a concentration threshold to observe this type of effect in polymers, right? So if you have a very low concentration, you don't observe this enhanced rigidity.
Okay, let me try to understand your question a little more. So you're thinking of stiff polymers, right? Or flexible polymers?
Stiff polymers, yeah.
Okay, and so you're saying you shouldn't observe this stiffening?
So in order, so you can observe this with only one chain, right?
With only one tree?
Yes.
Yes.
But you need a certain density of branches, right?
Yes. Okay, so you're thinking about the typical distance between two branching points, right?
Yes.
I mean, if you had just have a single line, you're essentially back to the okay. So the sheep's wool argument has it that, which I presented as exactly applying to our system, and it's not exactly the case. But the sheep's wool argument, which is a good first approximation, has it that the thing that matters is the total amount of length that you have inside your box. And it doesn't really matter how that length is arranged, because you just have those random crossings, as long as you're very under-coordinated, right? If you would be more coordinated than this, then the discussion would be different.
Yeah, but if I understood correctly, you need one branch to fall into another crossing point, right?
Yeah.
So this, when you release the stress, this get trapped here, right?
So if you do it after growth, it
yes.
may not be trapped. Your system might go back to, if I present my my simplest little diagram that I had at some point. It went back too far. I apologize. I think that's the diagram that I want to show here, right?
Yes,
so if I take this thing and I remove the compression from it, then those things may go back here, right? So the entanglement may not get trapped. But if after this, right, I have more growth happening, and this filament grows, and it grows a branch here that traps this branch, and then there's another branch that traps that branch, then you can't go back, right? Does that make sense?
That makes sense. Yeah, but it's my problem. It's not yours for sure. But
please go. Yeah.
I think that you need a certain amount of branches in order to see this, right? A certain concentration of activating sites that promote these branches in order to see this effect.
Perhaps a way to put it is that to the simplest order, the concentration of branching factors essentially determines how much length per unit volume you're going to have in your system.
Oh, I see. Yeah.
Right. So it does have an influence, but the major influence is through just simply how much polymer you're making.
Yeah. That makes sense. Yeah. Thank you very much, Martin.
Thanks.
Excellent. Thank you very much. In the interest of time, we have 1 minute. That's too short. Martin, I would like to thank you very much for giving your talk today in this unusual fashion.
We learned experience.
New experience for everyone. And I would like to invite our next speaker. I mean, usually would ask people to clap at this point, right? But I'm sure that
I'm sure they are, but they just muted their mic.
I'm sure that everybody is clapping worldwide. So I would like to invite Diego, Diogo, to come up.
I'll share my screen.
Share the screen. So this is the first of our two contributed talks coming from Lisbon. Are you actually in Lisbon? Right?
Yes.
Coming to us from the beautiful capital of Portugal. On tissues on pattern substrates. So we have 15 minutes for your talk and 5 minutes for discussion. So Diogo, please tell us about your work.
Yeah, thank you very much. And thank you to all the organizers for letting me talk about this project that we've been working on in CFDC in Lisbon. And yeah, it mostly focuses on the epithelial tissues on pattern substrate. And just for the motivation, I just want to make a simple parallel to inorganic materials, because when you talk about pattern substrates in soft matter, this has been a long-standing used method in this kind of problems of inorganic particles. And mostly are used to guide particles or even to constrain the final structures to get the more controlled final results in our project. And this has been the subject of many applications so far. But when we go to now the more recent research that has been done on living systems, although some things have already been applied, so we know that cells adapt to their environment, more specifically to patterns in the substrate, be it chemical gradients or mechanical differences. So we know that that's a relevant factor. So we can take some of the tools that we know from inorganic systems and apply them here in living to see if we have some results, because we can answer some problems of good fundamental interest from embryogenesis, morphogenesis, or even some more practical things in tissue engineering. So, but one thing that it's relevant to note is that the goals might not be the same, because inherently we are dealing with different systems. So for pattern substrates in inorganic materials, one of the goals can be to lower the bound of the substrate patterns given by lithographic techniques. So you can go to fewer, smaller and smaller length scales, and we have more control over the single particles. But when talking about living systems, there are some inherent properties to, for example, cells that might change these goals a little bit. So for example, here I have some experiments done with shape memory polymer substrate, where you can tune the pattern of the substrate with temperature and see how cells interact with a substrate. So by having a given pattern, the cells orient along those patterns, and then by changing the pattern, this triggers a biochemical response inside the cell that takes some time to develop. And so this gives the cell some inherent adaptation time, and thus they take some hours, sometimes hours, or even days to adapt depending on the change in the pattern itself. So there has been some things done in single cells, and what we've been looking at here is now let's take this inherent property of cells of adapting to the patterns and now putting in the concept of living tissues, so confluent tissues where cells are touching each other. So we also have these new mechanism, these two mechanisms, adaptation and on the other cell-cell interaction, and you want to ask some questions like what are the relevant time and length scales of these problems. So this is a purely theoretical study, so some numerical results and then also some analytical. And for the numerical, we use the self-propelled Voronoi model, which has been already introduced in the literature, and it's quite used in these living tissues problems, which, like the name says, it essentially uses the Voronoi tessellation of space to take the shapes of the cells. This is just a two-dimensional model, so we don't take into account the height of the cells. We approximate that it is negligible and that most of the dynamics comes on from the cross-sectional variations of the cells. So each cell is given area in a given perimeter, and each cell is constrained by this energy functional that I have here. So we have one term for the area, another term from the perimeter. So each cell essentially wants to have a given preferred area and a given preferred perimeter at each time step, which the area term takes into account the three-dimensional properties of the cell, and the perimeter, the adhesion and contractibility of the membrane. So using this inherently multi-body interaction, we can calculate the trajectories of the cells using this overdamped equation. I have again here the interaction term, and we just add a new term because obviously we're dealing with living systems. These systems are characterized by taking energy from their taking nutrients of their surrounding and transforming them into self-propelled motion. So we take one of the most simplest descriptions, this kind of activity of cells, which is just say that cells have a self-propelled velocity and go in a specific direction that is controlled in this way in order to give it a self-propelled motility that is persistent in time. So then this is the standard molecular dynamics loop, and we use this to study our living tissue. Just before going to the substrate part, I just want to focus on one key result that has been already known for these kind of models, because it will be helpful later on. So here I have a phase diagram of these two parameters. On one hand, the activity, and on the x-axis, I have this geometrical parameter, which is simply an adimensional parameter, which gives the ratio between the preferred parameter, here in the energy, and the square root of the preferred area. And it's just to say that depending on where we are in the phase diagram, we can actually be in a fluid, cells can be in a fluid-like state, so can diffuse more or less freely around the tissue, while if you are in this region, the solid-like regime, cells are constrained to move in their cages, so they are not able to move as well. So now for this substrate part, we considered one of the most simplest way to introduce this problem into this topic. And we just consider, okay, we have our simulation box. We just consider a pretty straightforward and simple pattern, just two half planes, left and right. And we say that cells on the left side want to have a given shape index, the geometrical parameter, and cells on the right want to have a different one. And the shape index that they want to have, this small p naught, depends on their position. So cells that are always on the right want to adapt to that substrate, let's say, and cells that are on the left want to adapt to the substrate that they are in. Every time they cross the substrate line or the borders, because we use periodic boundary condition, for example, a light green cell will change to a dark green. And it will change according to this expression here, a simple exponential decay, which we have here an adaptation time scale tau, which controls how long a cell will take to adapt after it changes the substrate. So the larger tau is, the more time they take to adapt. Okay, so with this model, we explore a couple situations. First, the fluid system. So like I said previously, both sides of the substrate now are in the fluid regime, and their cells are able to diffuse around and exchange from one side of the substrate to the other. Just one thing that I want to point out is that in this first simulation, we're considering that diffusion of both sides is very similar. So their geometry is very close. just so we can make a first initial approximation of what is happening and understand what are really the relevant things at play, and then we'll increase the difference between the two substrates and understand what differs. But first, for this case, what we see is that we initially have a fully segregated state. So dark green on one side, light green on the other. And as we increase this tau, these adaptation timescales, cells start to jump, well, start to diffuse from one point from one substrate to the other. And these shades of green start to appear, which are cells that are still adapting until we get the fully mixed tissue. To characterize this, we introduced the following order parameter, which is fairly straightforward. We here just have an Heaviside function. And the way we calculate it is here represented in this schematic. We go to a cell, for example, here in orange, and we ask how many of its neighbors have the same or have their shape index similar to the shape index of the orange cell, so within a given threshold. If the answer is all of them, then this demixing parameter equals 1 for this orange cell. If they're all different, the mixing parameter equals 0, and it can vary between 1 and 0, depending on the number of cells, depending on the number of neighbors are alike. So we average this throughout the whole tissue, and here we have how this parameter evolves with tau. So the mixing parameter initially for very small tau is equal to 1, approximately. And this is because cells adapt quite quickly, and then it starts decreasing until it reaches 0. And here I have a couple snapshots of these 3 different points just to illustrate the different stages, let's say, of this evolution. So for the color coding here, in green are cells that have the mixing parameter 1, in red the mixing parameter 0, and in blue are cells that have the mixing parameter in between. So initially you have 2 large green clusters, and this only for small taus. And then as we let tau increase, so will the red cluster until it spans the entire system. Furthermore, we also have here a new time scale that I still haven't talked about, this tau d, which is simply the linear scale of the size of our simulation box, squared divided by the diffusion coefficient of the cells in the tissue. And if you rescale this by the adaptation time scale, we can see that for different system sizes, we have all the curves collapsing. So this somewhat highlights the competition between these 2 relevant time scales that are at play. On one hand, the typical time it takes for a cell to adapt. On the other, the typical time it takes for a cell to diffuse a given length. So if the adaptation time is very quick, cells pretty much adapt too quickly to move the necessary length to change substrate, but as tau increases, they start to compete and mix more and more. So just to highlight a bit more this competition between these 2 time scales, we proposed a continuum model to try to describe what is happening in the system. So you have this U, which is just a population of fully adapted cells. So cells that have the same geometry, so shape index as their substrate they are on already, so they're fully adapted. And we say that this population evolves in this way. So 2 things to consider is that we only consider the x-axis evolution, and this is because of the symmetry of our substrate, because you only have these 2 half planes which are symmetric. And furthermore, one thing that we also consider is we only consider the evolution of this population for only one of the half planes, because again, both half planes are very similar in properties. So if you discover a population of fully adapted cells evolves in one side, you have the same in the other. So we have the diffusion term. The diffusion term simply tells us that the cell diffuses around, and every time a cell crosses the substrate line, it is now adapting, so it leaves the population U. So that happens when it changes substrate, and it adapts with a certain time scale, and we just say that, okay, cells that are currently adapting, so in the population of 1 minus U, adapt with a specific rate, capital T. So we can solve this equation without going into much detail about these calculations. What we see is that, again, we recover the relevant timescale of the system, this tau d over tau, exemplified here. We just have this alpha parameter that we use to fit to the numerical results, which I have here on the left side in this plot. A good comparison between the two for different profiles of the of our simulations. Then we can do the same for the demixing parameter. And here the demixing parameter, we just make a simple mean field approximation. And we see here in this black line, we plot this result within the same points that we took for the simulations, and we see that both are also in agreement. So now just to finish, if I don't have that much time, I'm just gonna go through the solid fluid phase that we also studied. And here the difference is that since the solid phase is not very mobile, so the solid particles cannot diffuse a lot, they're constrained by their cages, we actually, as we increase tau, although the fluid side mixes, the solid side remains constrained. And if you make the same analysis, we see that on one hand, the fluid side still has the same properties as before, but the solid one breaks this because it is no longer diffusing. So just when in my conclusions, I just want to say that, okay, we have proposed this type of study for the living tissues on pattern substrate, and we also propose a mixing length scale that can be used using experimentally relevant parameters to try and calculate what is the minimum length scale necessary for mixing. So we hope our results have some implications on this type of problem. And also acknowledge financial institutions and my collaborators, and thank you all for your attention.
Thank you very much for your talk. We have room for, depending on length, 1 or 2 questions. Let me start by asking. So you, this is something I'm not sure if I completely got that part, but so you're looking at initially at the liquid-liquid system, and then the difference is simply that between the dark green and the light green is simply that they have a different substrate, but not that they have different ratios of their area to perimeter, right?
So the idea is that they change slightly. So they are both in the fluid-like regime, but they change slightly just enough that we can produce differences, we can measure the adaptation between the two sides. But we make those differences small so that it doesn't play a significant role in this first analysis of the system. So you can say the geometry of cells in the left and on the right is somewhat different, but we can approximately say that they don't play a relevant role in the dynamics of the tissue. It's only really looking at adaptation and how it influences the mixture.
So we have a question in the chat, but to say my connecting question to this is, does this difference, is it then so small that you don't have an effective surface tension between your two liquids?
No, yeah, we don't have any surface tension at all between the two liquids. So in this segregated state, there is no surface tension present. It is just the fact that since cells adapt so fast, they're not able to go further enough into the substrate to start mixing, as in this case down here. So yeah, there is no surface tension. The only thing that separates these two is purely dynamical, that they just don't have adapt very quickly.
All right, thank you. And then the question in the chat, you probably also see it, is simply, could you remind people what the definition of the parameter U is?
U. This U is the population of cells that are fully adapted. So in essence, for example, it's here all these cells that are dark green, so that are fully adapted to the side of the substrate that they are in already.
All right, I hope this answers people's questions. Then in the interest of time, if there's a very short question, I can allow it. If not, I would like to thank you for your talk and invite your, I think your lab mate to come up. Igor?
Yes, I'm here.
Morning. You're
okay,
also
good morning.
You're also in Lisbon?
Yes, I'm in Lisbon right now.
All right. Connect nicely.
Okay, let me see. How do I share my screen?
So following on the previous talk, which was about adaptation of cells, you're going to talk about proliferation of cells, if I'm not mistaken.
Yes.
And so I guess we will have cells that now also divide.
Yes.
Thank you. So we are all ears for the next 15 minutes.
Okay. So everybody see my presentation and my mouse pointer?
Yeah.
Okay. So I first have to thank the organization for this amazing meeting that we are having, and also for the opportunity to show this project. This project is actually an ongoing project. So if you have any suggestion or question, maybe if you think later about something, you can send me an email or maybe reach me on Twitter. So this project is, so we have proliferation. So what we want is to go from a single cell to a tissue. So what we want to see is how you can go from those two different scales. So the scales, as the first scale is the scale of a cell. A cell by itself is a really rich system, and can cite an infinite number of amazing works about the movement of cells on substrates, and you can study the movement of a cell in a flat free substrate. You can study in a constrained environment. You can study the movement of cells with obstacles. And on the other side, on the other scale, you have a tissue, which is also a really rich system to study. You can study phase transitions and relate this with cancer metastases. You can study tissues on patterned substrate, as Diogo previously presented to us. And you can also study wood healing. But here, what I want to know is how you go from one cell in a flat substrate, how the cells proliferate and try to reach confluence on that substrate. So the project starts with an experiment. So what we have on the experiment is culture of glioblastoma multiform cells. And what basically we have is a solution where the cells are on the solution, and they are quite inactive when they are on the solution. In order to proliferate, to interact, and to multiply, usually they need to be attached to the substrate. So what you have is, you have a series of wheels where you put this solution with the cells, and you let them rest in order that the cells by gravity will find the bottom of the wheel, and then they will start to doing their magic. So they will proliferate, they will interact. And what we want to see is how do they proliferate and they occupy this flat substrate. So we have a couple, so after some different times for 3 hours, 8, 12, and 24 hours, we fixed the cells on the bottom of the wheel, and we take some pictures. And for instance, here I show a picture for 24 hours. In blue, we have the cell nucleus, and in red, we have the cytoplasm. So just to have a look, here I show also what we have for the dynamics. We have for 3, 8, 12, and 24 hours. So with those images, we can now extract the blue channel in order to, by using image processing, determine what is the position and the typical size of each cell nucleus. So by that, we assume, right now, we're gonna assume that we have the positions of the nucleus, so we have the position of the cell. So we're gonna call position of the cell. So from this data set that we have with a set of positions of cells in a flat substrate for different time steps, we can now try to understand what is the spatial distribution on the substrate and how does this spatial distribution evolves in time. In order to do that, we use a measure of spatial correlation. We use the Ripple K function, which is defined as the average number of cells that you find inside a circle with, inside a disk with radius r. And you normalize this measure by divided by the cell density. This K function is connected with the pair correlation, with the most used pair correlation function. And when we measure this K function, we want to compare with something that we know, with a baseline. And typically what you do is you take the homogeneous Poisson process that we know that this K function needs to be equals to pi r squared. So for a completely uncorrelated distribution of points, you have this functional dependence. So what we found on the experiments is that we do not plot here K, we plot K over R squared because it's easy to compare with the baseline, which now is pi. So if you have the correlations, spatial correlations equals to pi, so you have a random distribution of points. In our case, a random distribution of cells. Here we plot against the normalized distance, where we divide it by the typical size radius of a nucleus. And here I show what is the evolution of this spatial correlation for 3, 8, 12, and 24 hours. We see that since we have a deviation from pi, which indicates that we have a correlation or a clustering around some specific values of the distance. And we see that this really show up for 3 hours, but as the time goes by, the correlation seems to decrease. So we can concentrate for a specific value of the distance and see how this correlation for that specific value goes with the number of cells that we have on the experiment. So we see that naturally this correlation decrease. In order to explain what we are seeing on the experiment, we designed a particle-based model with a couple of ingredients, physical ingredients. And the first one is adhesion. So in each time step, you try to add a new cell. Our cells here will be particles, circular particles. So you try to add a new particle at a random position of the system, but you do not allow superpositions. Another ingredient is division. So we want to have proliferation, so we need to have divisions. So with a probability lambda, and at each time step, all the cells can multiply, and so you duplicate the cell with probability lambda, and in the duplication you create a new cell with a distance 2 times the radius of your particle. But again, we do not accept superposition. We have mobility, so each cell can move in a Brownian motion with a diffusion coefficient D, and we have some type of cell-cell interaction where we allow superposition, we allow overlap when they are moving, but if we have overlap between 2 particles, we decrease the diffusion coefficient by multiply by a parameter tau, which is a number below 1. What are the general properties of the model? So we can, for instance, see how the number of cells increase in function of time. If we take, for instance, tau equals to 0, we naturally have an increase on the number of cells, but it saturates to reaching a maximum packing fraction of the system. But as soon as you start to increase tau, the cells can move on top of each other. You can have more than 1 layer, and you create space, and you can add new cells by adsorption or by division. But we are interested to see how is the correlation, the spatial correlations goes with this set of different parameters that we have. And here I show a typical profile that we can can see from the simulations. So all simulations start with one cell, and you let the cells, one particle, and you let the particles duplicate and proliferate in the substrate. And when it reach a specific value that you want of particles, you take a picture and measure the spatial distribution. Here I want to show for different values of the parameter lambda, and I want to focus the attention by the fact that you have a typical peak around 2. This is due to the fact that the duplication naturally creates a correlation on the system. So when you duplicate, you have a preferential position that the cells will take in the dynamics. But we are interested to compare with the experiment. So we simulate for the evolution for different number, the spatial correlations for different number of particles, and we see that similar to what we saw in the experiment, the correlation it's large and have a peak when when the number of particles is low, but as we increase the number of particles, the correlations start to go to pi. But we want not only a qualitative, but want a quantitative good agreement with the experiment. So we try to fit the experiments for 3 hours to see what is the best option, best set of parameters. So here we concentrate our attention to 2 parameters, the lambda and the tau. And since we want to fit for 3 hours, we are using the average number of cells that we have on the experiment for 3 hours as a set of as a number of particles that we have on the experiment. And we compare, so we let the system evolve. When they reach this number of particles, we take a picture, calculate the spatial distribution, compare with the experiment, and then change lambda and tau and see if we have an optimal parameter that explain the spatial distribution for that specific time. We see that we have a really good agreement for 3 hours. For that set, we have a minimum, we have an optimal set of lambda and tau. And however, we want to know is that, okay, we know what is the optimal for 3 hours. If I let my simulations run a bit more, I will have more particles. I can compare with 8, 12, and 24 hours. For using the set of lambda and tau optimal for 3 hours, I extend my simulations, and I show here in dashed line that the model captured the overall decay on the correlations, but it overestimates the correlation for all times above 8 hours. So that let us believe that the optimal parameters for 3 hours is not necessarily the optimal parameter for 8 and 12. So probably during the dynamics, we're gonna have to change both parameters, maybe one parameter, in order to correctly describe the experiment. So we decided to keep constant lambda, which is the duplication rate, and change the cell-cell interaction. And we changed the cell-cell interaction by increasing tau until it saturates to 1. And this increase is controlled by a new parameter called T. And we can see on this plot that so for T equals to 0, we recover the last result. So we have not a good agreement with the experiment, but as we increase T, we start to get a really good agreement with the experiments, which are this gray circles. So if I take this value of T equals to 0.1 and plot the overall spatial distributions, the overall spatial correlations for my simulations, I could compare with the experiment and see if I have a good agreement. One thing that is important to note is that this T value that I found that explains this decaying of the correlation for increasing of number of cells, it's actually quite high, in the sense that what is happening is that after a bit more of 3 hours or a bit more of 300 particles on my system, you basically go to tau equals to 1, and the cells start to not see each other, start to move on top of each other without interaction. So what are my conclusions? So by control this cell-cell adhesion, cell-cell interaction, and proliferation rates, we are able to reproduce the evolution of this two-dimensional spatial heterogeneous distributions and provide some insight about the underlying dynamics. We understand how the cells goes from this scattered distribution to a mechanically robust tissue, poses a technological and theoretical challenge to study morphogenesis, wood healing, and cancer. And several of these theoretical challenges is deeply related with important problems of soft condensed matter, such as social and no equilibrium dynamics. There is several questions that we have to answer. One of them is, for instance, why tau goes so fast? This is an artifact of the simulation, or does this have some biological meaning? One possibility that we are thinking about is the fact that since we are dealing with cancer cells, we know that there is a fact that cancer cells have no contact inhibition, in the sense that the normal cells usually try to form monolayers, but when you have cancer cells, you can have the cells can grow on top of each other on those type of systems. But this is just a hypothesis that we have to test probably with more experiments. So just want to finish to thank all, and if someone have any question.
Thank you very much for your talk. And indeed, there is room for questions. 5 minutes, because you ended nicely on time. And yes, I see Laura has a question for us.
Hi, thank you, beautiful talk. I just wanted to ask if you have think on including in your model the polydispersity in size of the cells, and especially during proliferation, that when the cell divides into two, the two daughter cells will have half of the surface area than the mother, something like that?
Yes, we actually started trying to model flexible cells. So that would be a next step would be maybe use different size of cells. But the problem is that we try to keep the number of the number of parameters in the system low, in the sense, and also parameters that we could kind of measure and compare with the experiments. But it's a good point. That's something that we can explore. Maybe try to use this polymerization of size, but not yet. We didn't try yet.
Thank you.
Thank you. I see Martin also has a question. Martin, can you just ask the question? It's more interactive.
Okay. Yeah.
Yeah. So I was wondering. So you use your model as a way to analyze experimental data and extract parameters from them. And sometimes when we do this, there's some of the parameters that we fit that we're very confident are at this value and very robust, and some of them are perhaps more dependent on the statistical realization that we're looking at. So I was wondering if you could, from your experience, communicate us the feeling of which parameters you think are the most robust that you determine, right? I mean. Yeah.
Okay. Yes. In the model, we have a kind of four parameters that we have to adjust. We have a fifth parameter, which is the radius, but we can extract this directly from the experiment. But we are there is the rate where you have adsorption, so you add new cells on the system, not by the duplication, but by taking from the bulk. We believe that this parameter only control the time scale. So we have a simulation this will probably, if I change this beta, I will only take more time on my simulation, less time. I was thinking also about the diffusion coefficient. I didn't show anything about the diffusion coefficient here. I assume is a value that was similar with some experiments that I found. But we also know that probably if I change too much, so if I put too much movement on the particles, they will mix and they will destroy this correlation created by the duplication. But we are pretty confident about this beta that they have some impact only on the time scale, and on the lambda. We also try to, instead of take a specific function for tau, we try to do this whole landscape for each time step. So we try to all possibilities of lambda and try to minimize the error, and we saw that lambda almost didn't change. That's why we later assumed that lambda didn't change. But the big
division rate, right?
The division rate, yes. The point is, the division rate naturally changed because as soon as you have more particles, since I have a rule on my model that I do not allow a new particle to come up on top of the other, in practice the division rate changes, but not by myself tuning it. But the big question is why this tau needs to increase. One possibility is this: as soon as the cells start to proliferate on the system, they help each other by probably produce some extracellular matrix to move with less work. I don't know. Actually, we are studying that.
Okay, thanks.
Thank you very much. And with that, we have reached the end of our session of this morning. I would like to thank especially our speakers from the second half. Also, of course, speakers from the first half, but they were thanked already. Martin, Diogo, and Igor, thank you very much for your nice talks. Thanks everybody who was here in this session with us for your questions and your presence. And we will continue with our second day tomorrow morning at 9:30. Again with invited and contributed talks. So thank you very much and hope to see you tomorrow. I think Laura is trying to say something with her microphone.
No, thank you. Yes, thank you.
Thank you very much.